Thank you for joining the Once Changing the World, India's first Future Tech Meets Sustainability podcast. And today, I'm delighted and honored to have with me Dr. Roman Bauer, who is a senior lecturer in the Department of Computer Science at the University of Surrey. He leads the Interdisciplinary Computational Modeling of Biological Dynamics and Biological Engineering Lab. Dr. Bauer's research focuses on neuroinformatics, AI, cancer research, and the computational modeling and analysis of biological dynamics, particularly the brain. He's also involved in the Biodynamo collaboration, which focuses on dynamic biological systems modeling. In October 2024, Dr. Bauer presented a simulation that mimics neuronal growth in the brain, which could have significant implications for understanding age-related conditions and developing future treatment for neurological diseases. So, Doctor, really appreciate you taking time and being part of the podcast. I think it'll be great if you could start with this computer simulation on how the brain grows neurons and its primary objective.
Yes, hello. Thanks, Eddie. Thanks also for having me on your podcast. Yes, so as you said, we simulate development of neurons and brain tissue, and we are interested essentially in creating computational models that help us gain insight into how neurons in nature develop. So we really simulate how the genetic code instructs neurons to grow out their neurites. So neurons have so-called axons and dendrites, and axons and dendrites allow neurons to communicate with one another via electrical signals. And so we are interested in simulating how do neurons gain their final morphology, and then also how they communicate with one another and how they learn, because neurons are highly plastic entities. So neurons are developing based on experience, and the experience not only shapes the synapses, so the connections between the individual neurons, but it also shapes the structure and morphology of the neurons themselves. And neurons are extremely complex. So current AI models are much simpler than the neural networks of the brain. And we think and we believe that if we better understand how the brain develops, then this has many implications, not only for AI, but also for medical applications.
Our brain, it's extremely complex. These neurons, we've got 80 billion neurons, 100 trillion synapses, the way they fire and wire, it brings about all of five senses: sight, sound, touch, smell, and taste. While doing the simulation, what's been your learning? What's been the core learning? And you say that it would help in age-related conditions and neurological diseases. Can you elaborate on how this will help for this interventions, neurological disorders and age-related conditions?
Yes, very good question. So there are two parts here, right? The first one is how do they learn? And then second, what are the biomedical implications for neurodegenerative diseases? So with regard to the first part, you can see learning as a highly multifaceted thing. So it's not a very homogeneous thing. Learning takes place on many different levels. I mean, the cells and neurons, they can change their intracellular properties. They can change so-called membrane receptors density on the neurons. So different kinds of neurons have different sensitivities to stimulation and to certain substances. And this depends on the distribution of those receptors on the neurons. And this is something that can be influenced by experience. But it's not only experience of the type of external experience, like when we see something, when we see an object, or when we hear a sound, or where we feel a sensation on the skin and so on. There is also experience that is self-generated, and we actually see that during development a lot. For example, there are so-called retinal waves. So these are waves in the neural tissue at the rear of the eye. This is a tissue that essentially allowing us to see, because it translates the visual input into electrical stimulation. But even before we see, so while we are in the womb of our mother, we can actually have impressions or inputs from the retina, which are generated by the retina itself. So it's not like there's some external input. It generates by itself so-called patterned activity, like retinal waves. And those retinal waves shape how the neurons develop. And by developing, again, I mean something very complex, because it's also the neuronal morphology. So how do the neurons look? As I said before, neurons have axons and dendrites, but the exact morphology of those axons and dendrites depends from neuron to neuron. It's actually, there are no two neurons that are exactly the same. There is always small differences, right? And we are trying to better understand, why do they look like this? Because I'm convinced that this is actually a key to understanding the brain. Is we need to understand how the brain gains its structure and its morphology, because the brain is extremely complex. It has almost 100 billion neurons, and it has 10 to the 15 synapses approximately. So this is a 1 with 15 zeros behind. It's a huge number. And if we want to understand, or at least gain some insight into this complexity, I think we need to understand how this complexity develops, right? Because the complexity of the genetic code is much simpler than the complexity of the brain. If you look at the genetic code, the human genetic code, it has only about 20,000 protein expressing genes, and so it's much simpler than the complexity of the anatomy of the brain. And so the question that we ask is, how does any kind of input, whether it's self-generated, whether it's coming from the external environment, or whether it's generated by the genetic code, how does this lead to the different cell types and cell morphologies? There are actually about 1,000 cell types in the brain. So it's not just like excitatory or inhibitory, right? Or it's not just neurons and glia cell. It's very multifaceted. There are lots of different cell types in the brain, which brings me to the second part of your question about biomedical relevance. So there are some studies that show that one can inject stem cells into certain types of animals and brain regions that help with certain disorders. And this is a very exciting research direction. A biomedical research direction is about, can we repair the diseased brain in some way? For example, people with Parkinson's disease or epilepsy or Alzheimer's disease, because they have neural circuitry that is not working properly. And if we could potentially, that's the idea, would be if you can inject neurons, maybe in their stem cells state, maybe they could integrate into the brain, and they could help the brain become again younger and almost rejuvenate parts of the brain. So that would be the idea of this kind of approaches would be to replace and regenerate and essentially make the circuitry healthy again. Now, this sounds all good in theory. However, it is really a complex task, because, as I just said before, we have thousands of different cell types in the brain. So you cannot just expect that you inject the stem cell and it will exactly become the right type of neuron in the brain, and it will exactly integrate as it should be in the healthy brain. That's not how this can work, right? Because during development, there are different stages. And during those stages, there are certain types of chemicals expressed, for instance, or different types of genes that are expressed at very specific times. And we need to understand at what times and why do those express themselves exactly at that time if we want to repair the brain, right? So that's why repairing the brain relies on understanding the development of the brain. Otherwise, there's no way that you can just expect to inject some stem cell and it will exactly by magic do what you want it to do. We really need to understand this developmental pattern if we want to repair it. And computer simulations are an excellent way to research this direction, because if we just want to create a model, we could, in principle, I mean, someone might say, like a devil's advocate may say, why don't you just do experiments? Why do you need computers, right? That could be an objection from someone. The issue is, again, has to do with complexity and also with data. We have now a lot of data available from different kinds of measurements of the brain. We have, for example, anatomical data, right, where we see the structure of the brain. We even have anatomical data during development. There are various different types of data sets where we can see how the structure of the brain changes over time, even before birth. And so, given this huge amount of data that we can now fortunately access, we need to make sense out of it, right? And just looking at the data would not be enough to do this. So we need computers to integrate all of those data sets in order to create a model that explains them and that makes sense out of them. And often computers can be used and also to generate predictions. So this is, I think, a very important part of any kind of simulation or of any kind of computational model, is that it should generate predictions. What I mean by predictions is, given that I have some type of data and I have a computer simulation, ideally I should be able to say, okay, if my understanding is correct, then I would expect, if I do this kind of experiment, I would like to see, I would expect to see this happening. And so this is experimentally verifiable. And that's essentially the ultimate goal, I think, of computational modeling, is to generate predictions that then can be experimentally verified. And if they're correct, we can say, okay, we really understand this now much better than before. Now we can go to the next step and we can iteratively refine our computational models and make them much more detailed until we have a coherent model of brain development. And then that would have implications for biomedical applications as well as technology.
So what I understand from what you're saying is that we are starting to understand the brain, and computational modeling is one of the ways, but eventually making sense of the data is going to be key to possibly creating interventions. Like the brain, I mean, I think AI is also considered a black box. And I think there is work going on, interpretability, to kind of understand what AI does at the back end, because nobody kind of understands it. And I think the similar analogy kind of stays true for even the brain. Brain is also considered a black box of sorts. There are various approaches to kind of, maybe structurally and functionally map the brain, but we're still kind of far away from, understanding or completely mapping out a human brain. Though the works have been ongoing with C. elegans, which we have completely mapped. We've mapped out a fruit fly, and I think recently Harvard and Google mapped out a cubic millimeter of the human brain. What does all of these mean? Do you think some point in time we'll be able to completely structurally and functionally understand the human brain? And once if you think we will be able to reach that, what would be these predictions? You said generate predictions. What do you think would happen if someday we are able to kind of completely understand the human brain?
Yes, a very exciting question, I should say. So I think understanding the brain is by itself is a question that has different levels of depth, right? I mean, you can understand the brain kind of intuitively, or you can understand it quantitatively, for instance. So there are different levels of how we can understand the brain. I would say, or my claim would be that a single human would not be able to understand the brain in depth, because it's just too many things going on at the same time at different levels. It's a highly heterogeneous process. And so I think but if by understanding the brain you mean that we can potentially create something that is similar to the brain in a way that it works similarly, it maybe has similar performance in terms of understanding certain concepts and even performing certain tasks, then I think we are on the right track in terms of simple tasks or tasks that are concretely and specifically that you can describe. You can say, okay, this is the task. I want you to achieve this and this. No, then I think we are on the for that we are on the right track. But the human brain is not like that, right? It's not like you have each time a concrete goal and that's what you need to achieve. It even creates goals on itself, right? It's in a way it doesn't have a single trajectory. It depends on the context, what exactly the next goal is, and so I don't think that current AI is very similar to the human brain. And if we better understand the human brain, I think this would revolutionize AI further, because obviously I'm myself very impressed by what current AI can accomplish. I mean, ChatGPT and so on, and various other large language models have achieved really impressive performance for various tasks. And I think this is great. This is amazing. But obviously, as anyone who knows about brain or cognition and who knows about what the human mind can accomplish, will confirm there are still many things that current AI does not really reach, right? It does not really, I think, really understand concepts well, because what current AI methods usually do, like large language models and so on, is that they are statistical models, right? They predict the next word, for example, based on what it has learned, and it's not really a fundamental understanding of what it's doing. So there are different approaches in AI, right? There is like this kind of more statistical approaches or data-driven approaches, where you have some kind of model that is not really the same as for example the human brain, but it is very good at creating statistical models based on all the data that we feed it. And current large language models are very good at that. They're very good at creating models of huge numbers of data sets, and they use this data in order to learn and to adapt trillions of parameters, right? Current large language models are extremely, really large. So the word is, the name is definitely well suited for that. But the problem is that, for example, they are not sample efficient. What do I mean by sample efficient? They need a lot of data. And that is one of the big challenges now that large tech companies are facing. They wonder how can they improve current large language models performance even more, because most of the things they can do is that they need more data to do this, right? And this is now really becoming a bottleneck. And that's one huge bottleneck is data or sample efficiency. We are much better at that, right? I mean, I can show you I can tell you some rules of a game, and then you will know how to play this game. You don't need to see someone doing it thousands or 10,000s of times in front of you. You will just understand the concept, and then you can very easily from that generalize. And another big issue, which is in those times currently is a big issue, is energy efficiency. So current large language models require extreme amounts of energy, and this is of course bad for reasons of sustainability and just ethically it's a big issue. And the human brain just requires about 20 watts, right? It's like light bulb, essentially, and obviously that's the energy that it needs to perform when it has matured. And of course, it takes energy to grow a human brain, of course, but still, if you consider that, several orders of magnitude more energy is needed for current AI. And so one of the big promises of understanding how the human brain works is that we could not only improve sample efficiency, but we could also improve energy efficiency. And that would, of course, be a huge progress for mankind and humankind.
You mentioned the largest tech companies building these LLMs are stuck because there's the data problem, and also they are not energy efficient, and so they're facing blocks because of the data is being exhausted, and even the AI generated data is not helping. So, but yeah, I'm sure that if we go towards like a brain inspired AI where there'll be we'll be able to solve all of these problems and build possibly like better machines with which maybe what we're dreaming of or we're trying to build, these machines which have human level intellect. Would you be able to explain approximate Bayesian computation and the role that it's played in enhancing the accuracy of your simulation?
So we have recently we have published a paper where we have used Bayesian statistics to essentially pinpoint or infer growth parameters of neurons. So what does that mean? So as I mentioned at the beginning, neurons grow based on rules that are encoded in their genetic code, right? For example, neurons they have axons, and at the tip of the axon is the so-called growth cone, and the growth cone can sense chemicals that are near the neuron, and or at in its local environment. And so when the growth cone senses certain chemicals, it will know that these are guidance cues, so they tell the growth cone where to grow, for instance, right? So the axons they grow according to certain cues that are in its environment. So there is not really any global instructor that tells each neuron, oh, you have to grow there, or you have to grow to the left, and so on. That's not how it works, right? In biology, you have only local information exchange, which means that everything has to happen without a global instructor that tells each element what it should do. And there are a lot of advantages of this kind of approach as compared to how we humans usually build things. So we humans usually we have some kind of plan, some kind of template or blueprint. And then, for example, if we build a car, I'm not an expert in how to build cars, but you will have some kind of blueprint that tells, okay, here this part goes to this part, the wheel goes on this part, and so on, and so everything has a predefined location from the start, and it's very exact where it has to go. But that's not how the brain grows. Our genetic code doesn't tell each neuron how it has to look. Our genetic code doesn't tell each neuron how exactly it should, or to which other neurons it has to connect. That would not be possible because there's not enough information in our genetic code to do that. So what we did in this paper, where we have used this Bayesian statistics approach, is we said, okay, let's assume that we know that we know the end result of a neuron. So there's a lot of data about how neurons look, right? So people have collected data that shows the morphologies of different types of neurons, and so there are databases that are public, like the Neuromorpho database, for example, that we used for this recent study that was published in the Journal of Mathematical Biology, and there. We said, okay, let's assume we have the end result. Can we infer what was the growth rule that led to this end result, so to say. So this tells us a lot of things, because it tells us essentially a rule for how neurons grow, right? It's a simplified rule, obviously. It's not the final rule that really is taking place, but it at least brings us closer to the truth. And using Bayesian statistics, we have been able to infer rules and parameters for those rules based on the morphology. And that goes into the direction of what you just said before about interpretable or explainable AI, because now we have an explainable model that tells us how certain neurons grow. There are other people, other labs that just see can they grow neurons that look the same way, right? That's another approach. That's not what we're doing. We really want to understand mechanistically how do neurons grow, because if you understand this, then you understand, for example, how you could potentially make a neuron grow in a certain way to, for example, treat Alzheimer's disease or Parkinson's disease, right? If we don't know how those neurons grow in nature, we don't really know how we can play with them or instruct them to generate a certain morphology. So it's really that's the key of it. And using Bayesian statistics, which is a very powerful statistical approach, we could indeed show that we can generate very plausible, very accurate neuronal morphologies, at least in simulation. And I would love to explore this further, and also create ideally collaborations with experimental researchers, so that we can have this loop between theory and practice, because that's very important for computation models, right? Is that we need to create predictions, as I said before, which then should be tested, and they will generate new kinds of data, which then can be fed into the computational model. So yeah, that's a brief summary of what we did.
Right. Interesting, very interesting. Doctor, was there any specific reason why you use the hippocampus region to grow this neuron? And will be great if you could also share your experience in developing the biodynamo software and its significance in the research.
Yes. So there was not any strong reason why this was used. I suggested other neuronal types, and then Tobias suggested those, and he preferred to simulate those. That I don't have a strong opinion on which neuronal morphology to grow. In both cases, the questions that we ask are the same, right? How does the final morphology result? In some cases, when there are connections that are very long range, then we need to increase the complexity of the model. So there are some reasons why one type of neuron may be preferable for an initial investigation than another type of neuron. But ultimately, this is something that can be applied to any kind of neuronal type. So it doesn't really matter whether it's a hippocampal or a layer 2-3 cortical pyramidal cell, for example, and so on. There is data in both cases, fortunately, and so I think this is really something that also needs more research, because there are not so many people at the moment who work on this, and that's why we are very interested, of course, in connecting with other researchers who want to collaborate with us, and so that we can step by step create more and more models that explain different kinds of cell types. And that would be my goal, so that ultimately we can bring it all together and create, at the beginning, maybe just a single brain area from the cortex, like visual cortex, for example. We could potentially simulate it already now if we had enough people to do that, right? So that's one part. And then regarding the simulation part, this is a big challenge still, is how do you exactly computationally model that? What kind of implementations do you use for such simulations? And I have about a decade ago, actually more than a decade, we started working on the so-called biodynamo collaboration. So biodynamo is an international consortium, and we have come together to work on a computational biology approach to simulate brain development. That was our initial goal, and we have then also used this for other types of applications, like for example cancer. But our initial goal was to implement a software that can allow to grow neurons and neural tissue, and ultimately an entire brain, from a single precursor cell, because we all stem from a single precursor cell, right? The zygote, this is an interesting concept by itself from an academic perspective. Is how does a single cell generate a brain, right? But then also from a computer science perspective, this is a very interesting concept, because if you want to simulate this, you cannot just write your own Python code quickly and look how it develops. You need to have custom software that is highly efficient. And that is the reason why we came together, because this is a highly interdisciplinary collaboration. So there are people who are experts in high performance computing. There are people who are experts in computational neuroscience, like myself, for instance. There are people who are experts in oncology. There are people who are experts in agent-based modeling, for instance. That is the approach that we're using. So this approach is called agent-based modeling, because it assumes that we have elements that are autonomous. So autonomous in the way that there is no global supervisor that tells each element what it should do, but it just follows its internal rules, which is obviously what I just explained before, the same as what is happening in cells, in biology, where each cell just follows its genetic rules. And so what we did is we managed and successfully wrote a highly performing agent-based simulator, which is called biodynamo, like the collaboration. And biodynamo is available as open source software. So it's a software that everyone can download for free. And also you could even potentially commercialize it, because it's under the Apache 2 license. So it's very friendly for commercial purposes as well. And so we have managed to provide this as open source. And since then we have grown a lot, and also companies have joined us. And we have now used biodynamo in many academic publications, but also in teaching and education. So we use biodynamo to teach students, for example, computational biology topics. And yeah, so far it has been really a very exciting journey.
Right. Doctor, you also have done work in the space of cryopreservation. I had interviewed a few folks in the space of cryopreservation. I had interviewed James Arrowwood from Alcor, and also Daniela Medvedev from a Russian company called Cryorus. What is the current state of technology when it comes to cryopreservation stands? With this computational modeling of cryopreservation, how do you think you could accelerate this technology? I mean, would love to hear your thoughts on that.
Yes. So at first glance, these two topics seem not very related, like brain development and cryopreservation. But actually they are, because cryopreservation is also a very complex process that is relevant to different scales, spatial scales and temporal scales. So in cryopreservation, what people do is they have some kind of biological material, like cells in suspension, or some kind of tissue, can be even brain tissue, right? And they're going to preserve it, meaning that they want to reduce the metabolic rate so that they can keep it ideally for as long as they wish for. And obviously this would have very, very far-reaching implications if we could do this for large tissues, including neural tissues. for biomedical purposes, but also for space travel, for instance, and so on, because traveling to Mars, for example, will take more than 6 months. So we cannot just expect that people will stay for 6 months for a long time in a confined space. That this would be psychologically very problematic, and so we need to think of potential ways to address these issues. And preservation or biostasis and cryopreservation are interesting because they offer a lot of solutions for many problems, including, for example, the current lack of or there's not enough organs that people can use for transplantation. Right, there's a long waiting list in all countries for organ transplants, and so. And if we could cryopreserve those organs, then this would be huge. This would save many people's lives. However, one challenge is, as I said, is this complexity, because if you have some tissue that you want to cryopreserve, then you need to first add some kind of chemical, some kind of so-called cryoprotective agent, which protects from injury during cryopreservation, because cryopreservation usually is deadly for cells, because the ice crystals can disrupt membranes or intracellular organelles and so on, and so the question becomes, how can we use those chemicals, but then also at what concentrations should we add those chemicals, because those chemicals are usually toxic, and so we cannot just pump as much as possible of those chemicals into the tissue. We need to be very careful, and also it depends on the temperature at which we add the chemicals, because if the temperature is high and we add those chemicals, then the good thing is that it will diffuse well, right? The diffusion constant is related to the temperature, positively correlated. So we have the higher the temperature, the more it will diffuse, and so it will protect the tissue overall better, spatially or volumetrically. But the problem is that toxicity also correlates with temperature, so it's more toxic at higher temperatures. And so if we reduce the temperature and we have chemicals that we add, it is less toxic, but it will not efficiently protect the tissue anymore, because it just doesn't diffuse well, right? So there are many questions. What are the concentrations to use? What kind of cryoprotective agents should we use? Because there are many different cryoprotective agents. None of them are perfect. They have all issues and challenges. Some of them are acting intracellularly, some are extracellularly. And also, how quickly do we cool down the tissue? Like, there are different ways of cooling down tissues and so on. So what I'm saying is, I don't want to go too much into detail here, but what I'm saying is that cryopreservation is a very complex field and discipline. And this has not been appreciated, I think, long enough in the scientific domain. Usually people think that cryopreservation is just a tool, you just have your cells and you cryopreserve it, and it should work or not. But not many people are really trying to improve cryopreservation, especially from a computational perspective, because, as I said before, computational approaches or models are important if you have very complex data, right? Because no human brain can alone grasp all those different factors. So what we did with BioDynamo is we created a software called CryoDynamo, and there we can actually model how temperature dynamics affect tissue over time, because BioDynamo and CryoDynamo are multi-scale modeling software tools. So they can model tissues at different scales. And so I want to bring in a computational component into cryopreservation research, or more of it. There are people, of course, who use computational methods, but there's not a lot of it. But I think if we could better model cryopreservation with computers, we could really very much improve cryopreservation. Because at the moment, it's mainly an experimental discipline, right? So people, they use some kind of cryoprotective agents and they see what happens. Does it work well or not? And it's more of a trial and error process. And there is, for example, no way at the moment to design cryoprotective agents, right? Ideally, if we could somehow say, okay, based on the molecular structure for this kind of tissue, we should use this kind of cryoprotective agent, that would revolutionize cryopreservation. But we are still not there, and I think we need advances in computational modeling of the entire cryopreservation process. And this has applications for many different domains, like you said before, including then also organ preservation.
Besides these, these cryopreservation protocols, computational modeling, has that been like, have you partnered with any startups who are in the space of cryogenic or preserving cryo organs? And what are your timelines? I mean, when do you think that we will, once we cryopreserve possibly these organs, if we get to the right protocols, do you think there will be like a technique where we could be someday be able to revive these organs or these cryopreserved bodies?
Yeah, these are all good questions. So regarding the first question, so we have written a manuscript, and it's almost ready for submission, where we show that we can actually improve or use computational optimization in order to improve the result or the viability of cells cryopreserved after slow cooling. And so I'm looking forward to hopefully submitting this soon. And also we have actually created a company recently, about a year ago, where we want to employ such computational models and optimization methods to revolutionize cryopreservation using AI. The company is called Oxford Cryotechnology. We have a website, so if people are interested, please have a look and get in touch. And we would like to use this kind of methods. Obviously, we need to generate data for this. So this is our currently our main challenge, is to generate data that then can inform the computational models in order to improve cryopreservation capabilities, potentially even leading to new kinds of cryoprotective agents. Because at the moment we are still using cryoprotective agents that have been around for quite a while, and we are not really able to design computationally such cryoprotective agents, even though in biomedicine now there are more and more drugs becoming available, and some of them already are in the market, that have been designed with computers. So I would like to follow a similar approach for cryopreservation, where we use AI to design new cryoprotective agents. And so indeed, this is definitely something we are looking to pursue. And regarding your other question, what was the second question? Sorry.
Reviving.
Oh yes, reviving. So this depends to a large extent also on the quality of after preserving. So when it's in the cooled state, so there are different factors, of course. I mean, one is can you lower the temperature without inflicting too much damage? I mean, there can be some damage because, fortunately, biological systems are often quite robust, right? So it wouldn't be a huge issue if some percentage is damaged after cryopreservation, but obviously it should not reach a certain threshold, and that threshold may depend on what kind of tissue it exactly is and what type of injury, of course, it is. And then also the thawing process is something that can lead to damage. And a collaborator of ours, Professor Ramon Risco at the University of Sevilla, he's working on rewarming, for example, so that the rewarming process can be more homogenized, because if the rewarming happens only at certain locations, then this can lead to pressures and cracks and so on. But ultimately, I don't think there is a one issue that would say, okay, it's not possible to revive after cryopreservation. I think in principle it is possible, but we need to improve. We need to do research, because at the moment, as I said, especially the computational component is not very advanced in cryopreservation. There are some limited studies which are very exciting, and there are also some very successful results. Just this year, there was a paper published where certain types of neural tissues could be cryopreserved, and they had some good viability after cryopreservation. However, those neural tissues are immature. They had to be immature, so it hasn't been shown that you can cryopreserve adult or fully grown neural tissue, and you can still retain the quality of that neural tissue after cryopreservation.
So besides this neuron simulation and cryopreservation, your work is also in, you mentioned, in the space of computational modeling of retina development, then self-organization of neurons, and AI for health. Is there any update that you would like to share from Combined Lab?
Yes, I mean, we have multiple lines of research going on that align with neural development. And for example, one student of mine has been looking into premature birth. So at the moment, premature birth is something that happens quite often now. I think it's more than 10% of all births are preterm birth. So they which is defined by birth before 37 weeks. And we know that there are certain alterations often in those preterm subjects in the brain. And we have created a model that captures well those alterations. And a student of mine is actually just now at a conference in Istanbul on complex networks. She will present this research. So this is a line of research that is very exciting, because it can again help us better understand what happens when the brain is born, or when a baby is born preterm. What happens in the brain, and potentially help us address those alterations, because often this can affect, of course, the life of individuals born preterm very much. And if we better understand what exactly is happening with regards to the connections, for example, that's what we looked at. We looked at the fiber tracts between brain regions in preterm born individuals. And we could show that we have a hypothesis that seems to work pretty well, that explains which connections are different and why they are different. And so this is one line of research we have looked at. Another line of research which was done with Biodynamo, and again we are almost getting to the end of that project. So we are finalizing this project and hopefully soon writing up a paper, is where we use Biodynamo to simulate an entire cortical column of a brain. So a mammalian brain has columns, so-called columnar structures that are repeated all over the cortex. And we have created a simulation that explains how this column grows. And this, of course, has again implications not only for AI, because if we can generate AI that also has this kind of organization, then we can arguably get closer to human type of AI, which has many advantages, right? It's more explainable, it's more relatable to biomedical applications and so on. And so we have done some work along those lines as well. And so, yeah, there are many exciting directions we are currently pursuing with regards to neural networks. Another PhD student of mine who is now also finalizing his project, and so I hope next year we'll have some really nice papers. Is a very exciting project on AI that is more efficient. So as I mentioned at the beginning, there are many parameters usually on those neural networks that need to be learned, and that's also one of the reasons why it takes so much energy to train them. And my student has been looking into creating AI or networks, neural networks, artificial neural networks that are much more, they have much fewer connections, and so they require less time, less training, and they require less energy to be trained. And also it looks like they can even perform sometimes better than networks that are much larger. And this is interesting because if you look at the human brain, for example, it's quite sparse in terms of connections, right? It's not like if you look at the neuron on this side, it is very likely to be connected to another neuron. No, they are connected via very large. Sometimes they are connected. Usually, they are not connected. In most cases, sometimes they're connected with large range axonal connections. But usually, the brain the brain is sparsely connected, and while current AI methods usually require a lot of connections, and so we have taken some inspiration from the way that the human brain is organized in order to create AI that is much more energy efficient, much more time efficient, and so this is another exciting research project that hopefully we will be able to publish soon.
Dr. My last question: what are the challenges you think, if you overcome, will accelerate your work and your research, and paint a picture of what the future is going to look like? Because you mentioned about, right now your work is on simulating a neuron. There are this team working on simulating an entire cortical column. Eventually when we go during the course of the conversation, we spoke about, simulating or the entire human brain. If you get to kind of that route, there are so many neurological diseases. Could you paint a picture of that future where we'll be able to possibly maybe take those neurological disease and sort these out, possibly?
Yes, very good question. So regarding the first part about the current bottlenecks or challenges, in my case, I think a bottleneck is still the computational one. So we have a lot of data, but we are limited in terms of creating computational models that can explain all of those different data, mainly because of performance. So as I mentioned, BioDynamo is very efficient. It can simulate billions of elements, but it's still quite far away from being able to simulate an entire brain, right? So what is important there, I think, is if we could use cloud computing. So if we could distribute BioDynamo simulations across hundreds, maybe thousands of servers around the world, then I think we could actually simulate an entire human brain in principle. And that would, of course, have far-reaching implications, as you said, for biomedical applications or technological applications. And so I think if we could address this, if we could somehow adapt BioDynamo so that it can use cloud computers, maybe even cell phones, right? We all have cell phones. Most people have cell phones that often are not used, right? If we could maybe distribute, if we could, in a kind of citizen science type of project, if we could use those idle computer hardware, whether it's on cell phones or some laptops that are maybe at home and are not completely used, that would be really a dream, because then we are not really very much limited anymore in terms of computational performance, and we could really scale up our models. As I mentioned before, my student is currently simulating an entire column, but this is still far away from an entire brain. And so we need, I think, support in creating computer infrastructure that can leverage scalable, ideally cloud computing or citizen science type of infrastructure. And that brings us to the second part of your question. So if we had this opportunity, and if we could then create models of an entire brain, this would lead to vast improvements. I will elaborate on the technological side of things. I mean, one really nice domain of AI research is in neuro AI research. which is this more biologically realistic approach, where we can more brain-like compute. And that means we could also compute with spikes. So that's a big difference what current AI is doing. In current AI, we don't have spikes. So we have artificial neurons that have analog value, but that's not how the brain works. The brain has neurons that spikes. So they have short action potentials, and these signals are then propagated to other neurons that then spike, and so on. And this is much more energy efficient, but it also has advantages with regards to inputs that are changing over time. I won't get too much into detail here, but there are certain computational advantages of using spikes, not only with regards to efficiency, but also with regards to time. And so if we could create a human brain model that has such spiking neurons, then we would have a completely different way of computing, right? We could generate new kinds of computers that are much more energy efficient and that are not centralized. So we could then also generate little spiking computers that are maybe like sensors and that are distributed elsewhere, and they already compute at the edge. So this is a field called edge computing. And if we have this kind of approach, if we can understand how the brain works, I think we would also make advances towards edge computing, because the brain is highly distributed, right? We have different parts of the brain have different roles. And if we understand how the brain achieves this compartmentalization of function, on this modularity of function, I think we could create a completely different type of computer that would vastly outperform any type of computer that we currently use, and it would be much more energy efficient and much more sustainable. And then with regards to the biomedical advance, as I said before, Alzheimer's disease, Parkinson's disease, various other age-related diseases could be addressed, because we would have essentially an in silico representation of the brain, or a model of the brain, and we could test different drugs on this brain. And obviously we still need to do experiments, but we could interact and we would speed up the entire drug development, because we have a three-dimensional representation of the brain. We have the right type of synaptic modeling and the right type of detail across different scales. So we could even test in simulation how will, if I, for example, in the simulation stimulate a certain part of the brain, how will this affect the whole network, right? What will it do? And we can create predictions, and that will allow us to test in the computer various different types of treatments before we do those treatments, so we can improve the efficacy of the whole development process of treatment. We have actually published just this year a simulation with BioDynamo where we simulate radiotherapy for cancer treatment. So this was not on the brain, but it's the same principle. We can actually use BioDynamo to optimize radiotherapy treatment so that we have less side effects that we don't want, and we can improve the outcome with those kind of simulations. So it's a very exciting approach to create whole organ simulations in 3 dimensions with BioDynamo, because it will allow us to much more effectively treat any kind of disease.
Right. Doctor, really, really appreciate you taking time and being part of the podcast. I hope there is, like you said, more compute, which enables for someday maybe in silico simulate the entire human brain. And if we get that, I mean, what a world that we would be getting into, to have like a digital copy of a version of the brain where you said, you test out, like if there's a certain problem, you test out a certain intervention, a drug, and see how it works, and okay, this is something which is working. I mean, that world would be so fantastic, because I guess today we've got so many problems, largely these neurological disorders. It leaves a human completely, it maims the human, completely. And I think if you are able to, somehow create a complete human brain model in silico, the things that we will be able to do would be fantastic. So more power to you and your entire team, and may all those that you wish, those infra, compute, GPU infra, maybe falls into the place somehow. So really wish you the best for your work. And to my listeners, if you like what you see in here, then please press the subscribe button. And until next time, see you guys. Bye-bye. Thank you, doctor. Really appreciate this. Thank you.
Thank you. Pleasure talking with you.
Yeah, thank you.